Hi, my name is Michele LeRoux, and I did my graduate work at the University of Washington in Seattle. And I'm going to tell you today about work I did in graduate school studying how bacteria interact with one another and how they survive out in the environment and in our bodies. So I'm going to start out by telling you that I really love yogurt. It's one of my favorite foods. I eat it almost every day. I even make my own yogurt now. But early on in graduate school, I was looking at one of those little yogurt containers where it says contains live and active cultures. And I wondered if that was really true. Would I really see bacteria in there? So I took a little bit of yogurt, I put it on a slide, and I went and looked at it under the microscope. And this is the picture I took. Look at all those bacteria. There's long rods, there's short rods, there's these little coccoid bacteria that are forming these chains. And actually different types of yogurt had slightly different bacterial groups in them that gave them different tastes. So this was really my first exciting glimpse into this invisible world of bacteria that are all around us. They live in these communities, and they make delicious things for us to eat. So this got me really excited about the idea of studying how bacteria live in these communities together. So of course, bacteria are not just in our yogurt. They're out in the environment, everywhere you look, basically, associated with plants, in the soil, in the water. There's bacteria in the ocean. Every teaspoon of seawater has about 5 million bacteria in it. So think about that the next time you accidentally swallow a little ocean water. It's a lot of bacteria you've just taken into your body. And then, as I mentioned before, there's bacteria in our foods. Cheese is another type of food that we require bacteria to make for us. And of course, there's bacteria in our bodies. In our guts, there's billions of bacteria. There's bacteria in our mouths and our skin. And a lot of them perform really important functions for us. They help us digest our food, and they help our immune systems work properly as well. But of course, bacteria can also cause really, really devastating infections. And that's actually usually what we've been studying them for in the past. So the organism that I'm going to tell you about today is this organism called Pseudomonas aeruginosa. And this is an organism that's actually mostly found out in the environment. It lives in the soil, lives in the water, and we come into contact with it all the time. It lives all over the place. So mostly it doesn't really cause us a lot of problems. But occasionally, Pseudomonas can get into a particular human environment, and it can really wreak havoc. So this is a little bit like a termite. You know, a termite, mostly it lives out in nature. It can build some really cool mounds. It will digest cellulose and wood, and it plays a really important role in the ecosystem. But if a termite gets into someone's home, into the wooden framework, and starts chewing up that wood, it can be really hard to get rid of, and it can bring the house down. So it's the same with Pseudomonas. You know, mostly we don't worry about it, but if Pseudomonas gets into the wrong human environment, it can be really devastating. So what environments are those? A lot of us might have loved ones, family members that are in nursing homes or confined to wheelchairs. And people like this are often prone to getting bed sores or chronic wounds. These are also common in people with type 2 diabetes. And these are wounds that just cannot heal. They just stay open wounds for sometimes years. And they can get infected with bacteria. So especially if Pseudomonas gets in there, it can be really, really bad for these people. It can get deep into the tissues, and it can be impossible to eradicate, and sometimes leads to amputations or even death. And we don't quite understand why it's so difficult to treat Pseudomonas infections when they get into these chronic wound environments. Another environment that Pseudomonas really thrives in are the lungs of patients with a genetic disease, cystic fibrosis. So these people have a mutation that causes them to have really thick mucus in their lungs, and this makes an ideal environment for this bacterium. In fact, 80% or more of people with cystic fibrosis will acquire Pseudomonas at some point in their lifetimes. And once they do, it's impossible to eradicate it most of the time. And it will go on to lead to the decline of their lung function and ultimately their deaths. So we really, really need to understand better how Pseudomonas is causing these problems and how we can treat these infections with Pseudomonas. So what I decided to do was to kind of step back from thinking about Pseudomonas interacting with humans directly and think more about what does Pseudomonas need to survive? What is motivating a lot of its behavior? How has it evolved over the years? So what I decided to do was think about putting myself in the shoes of this bacterium. Well, of course, not exactly in its shoes because it's a bacterium, it's a single-cell organism, it doesn't wear shoes, but thinking about what it needs to survive. So Pseudomonas needs, just like us, food to eat. it needs a place to live. It needs a home. And it will fight to the death to be able to make sure it maintains these resources. And actually, usually what it's fighting are other bacteria that it's competing with for its food in its home. So this is where it's really helpful to think about how long bacteria have been around and what's really been driving their evolution. So the Earth first evolved around 4.5, give or take, billion years ago. And not too long after that, in evolutionary terms, there's evidence of the first bacterial fossils. Sometime later, we have evidence of the first bacterial ecosystems. So these are where groups of bacteria live together and interact with one another. So this is where bacterial interactions would have first started evolving. Now, eukaryotes didn't show up on the scene until much, much later. And of course, humans have really only been around for the blink of an eye in terms of evolutionary time. So this green bar represents the amount of time that bacteria have been interacting with other bacteria. That green bar that you can barely almost see over there, that I'm showing you down here, is how long bacteria have had to interact with humans. So it's a much, much smaller amount of time. And yet that's what we usually end up studying in the lab. So I thought, well, there's probably a lot to be learned about bacterial behavior if we focus a little bit more on bacterial interactions with one another. And that might even inform how we think about their interactions with humans. So what I decided to do was to start by focusing on one particular bacterial weapon that it uses to compete with other organisms. And that is this secretion system called the Type Six Secretion System. You can think of it as a syringe that injects toxins from one cell into another. And this pathway was actually not discovered too long ago. And the lab that I did my graduate work in, the Michele lab, were one of the first to show that this pathway actually injects toxins from bacteria into other bacteria. So this is kind of what this might look like. If you have this green cell, let's say this is the Pseudomonas cell, it has these little red dots that are representing toxins. It will literally inject them into the cell next to it, which will cause these toxins will cause this other cell to die. And if we zoom in on this a little bit, you can see the syringe here I'm depicting as kind of a tube. But it's actually a molecular machine made up of a bunch of different proteins that are able to push toxins, from one cell into another cell. And these toxins have all kinds of different functions. We're still discovering new ones all the time. But for example, they might degrade the cell wall of the recipient bacterium, which would cause it to burst. They might attack the membrane of the cell. And some of them even go after molecules that are required for survival, such as RNA or DNA or energy molecules. And all of these things will cause the cell to die. So the next thing I want to do is show you what type 6 looks like in action. But before I can do that, I have to explain to you what to look for. So how do we watch cells dying in the lab? Well, what I like to do is I have cells that are expressing a colored protein, such as green fluorescent protein. They're making a lot of it, so they appear green when you look at them. But if that cell were to get a rupture in its membrane or in its cell wall, what will happen is that the green protein will flood out of the cell. And then after just a second or two, that cell will appear dark. So you're literally just going to see a green cell disappear. But really what's happening is that cell is bursting open and spewing out its guts into the surrounding area. And so just keep that in mind. When you see a cell go dark, really it's exploding in a actually pretty violent way. So let me show you what I'm talking about. So here we have Pseudomonas aeruginosa, and it's labeled in a red color. And we have a competitor organism that's labeled green. So you're going to be looking for these green cells to just disappear. So to make it a little easier to identify those, I had the software that I used to analyze this data outline them in white outlines so you can find them more easily with your eye. So just one note about this competitor organism. This is an organism that's called Burkholderia thailandensis. This is another bacterium that lives in the soil that would potentially interact with Pseudomonas in the environment. You don't have to remember the name. From now on, I'm just going to refer to it as the competitor. But one thing you should know about it is that it also has a Type 6 secretion system. And that will become relevant later on in my talk. So these systems are actually found in a lot of different bacteria. About 30% of bacteria that we've looked at so far have these secretion systems. So this is a common weapon that bacteria employ to fight with one another with. All right, so let's see what this system can do. I'm going to play you this movie of these two populations growing together. And hopefully what you can see is these green cells, the ones that are outlined in white, are just disappearing from view. And they're just popping as the cells die. So how do we know that this is because of the Type 6 system? Well, what I did next is I took the same competitor cells, but then I grew them with a Pseudomonas strain that did not have this Type 6 secretion system anymore. I inactivated it by deleting one of the key proteins. And now what you can see is the Pseudomonas cells are growing with this competitor. And I'm playing both movies at the same time so you can appreciate the difference. So now you see that there's actually not a lot of these green cells that are popping anymore because the Pseudomonas has lost its weapon. And so especially by the end of the movie, you can really appreciate how many more green cells there are in this movie where the Pseudomonas doesn't have its weapon compared to the one where it does. So this is what Pseudomonas can do. It can basically wipe out most of these competitor cells. But the question that I really had going into this was, what is the system itself doing? When does it get turned on? When does Pseudomonas know that it's time to mount this weapon and fight against a competitor? So let me show you one of the ways we can look at that. And that's by labeling one component of the system itself with a green fluorescent protein. So now we're not labeling the whole cell, but you can see these little dots forming in these cells. And each time you see one of those dots, that's actually the system coming together, and we think it corresponds to when it fires these toxins out of the cell. So what I've shown you here are just Pseudomonas growing by themselves, so they don't really have anyone to attack. And I've activated the system artificially, so we have something to look at, to study in the lab, because when you just look at the cells normally, there's not much going on. This kind of makes sense. These cells don't want to just be making all of this protein and firing it all out of the cell for no reason. That uses up a lot of their resources. And remember, I told you, bacteria are trying to survive. They don't have that many resources. So how do they know when is a good time to turn the system on so they can fight and actually have it mean something to them? So what I decided to do was take kind of a similar approach. I labeled one of the proteins of the Type 6 apparatus, and then what I did is I mixed Pseudomonas with this competitor and looked to see what happened to the system. So the first movie here that you can see over there is Pseudomonas just growing by itself. The movie on the other side is Pseudomonas that's growing with this competitor. Now in this case, I've labeled the competitor with red. I'm going to switch colors back and forth a little bit. We had to do that for some technical reasons, but I'll always make sure to tell you which population is which. And so I'm going to play these two movies simultaneously so you can appreciate the differences and look to see what happens with a green protein. So you see some dots forming, and then you see the cells get green. And hopefully what you can appreciate is that the cells growing with the competitor are a lot greener than the cells that that were just growing by themselves. And so what does this mean? What this means is that the Pseudomonas cells in the presence of a competitor are actually making more Type 6 protein. And not only that, they're actually assembling it more frequently into those foci, those dots that you see, which means that they're firing it more frequently. So this makes a lot of sense, actually, when you think about it. Pseudomonas somehow realizes that it's near a competitor, and it makes more of this weapon to fight it with. The next experiment I did, the result was a little bit more puzzling. So the first two movies I'm showing you here are the same ones I just showed you, Pseudomonas by itself, Pseudomonas with a competitor. But this last movie is Pseudomonas growing with the same competitor, but now, remember I told you that competitor also has a Type 6 secretion system? I've deleted the Type 6 secretion system from the competitor. So now let's look to see what happens. So the first two movies, you see the same thing. You see the cells with a competitor get really bright. But what you see here is the cells growing with a competitor that doesn't have its own Type 6 system. They don't turn on. They look just like Pseudomonas growing by itself. And we can see this in a graph form as well. So we quantified this data. And we're looking here at the levels of Type 6 protein over time. And you can see that when Pseudomonas grows by itself, you see a little bit of an increase. But when it grows with a competitor, that increase is much more. It's making more of this Type 6 protein. Now when the competitor doesn't have its own Type 6 secretion system, Pseudomonas doesn't turn its on either. So this was really puzzling. How is this working? How does Pseudomonas know the competitor is there, first of all? Then how does it know that it has this pathway as well? So this is a little bit of a silly example, but let's just imagine for a second that Pseudomonas is kind of like these Pseudomonas cells are kind of like a fleet of spaceships. So they're out in space. They're trying to figure out which other spaceships are attacking them, which ones are just kind of flying around and not bothering them. So let's say, so what I've just told you is that if it detects another spaceship that also has this weapon, it's going to activate its weapon and fire at it. So you can kind of think, if you think about a cell, how is it detecting that this other thing is there? These are single-celled organisms. They don't have eyes or ears or the senses that we have that we can see what's going on around us. So we wondered, well, maybe it's somehow sensing this syringe puncturing it on the outside of its cell, or maybe there's some other way it has of sensing some things going on around only when these other cells have a type 6 secretion system. So to distinguish between those two possibilities, what I next decided to do was to look to see if Pseudomonas needs to be touching this other organism. So the syringe can only attack Pseudomonas if the cells are directly contacting each other. So this is data from that same movie I showed you before where we looked at the type 6 secretion system turning on. But now we're just looking at one of the fields at the very end of the movie. Instead of looking at it in green, I've colored it so you can appreciate the differences in expression a little bit better. So if it's red, that means that there's more of the type 6 protein, and the bluer it is, the less there is of it. And what I've also done is I've highlighted which cells are contacting the competitor and which are not contacting competitor. So you can't see the competitor the way I'm displaying it here. You'll just have to take my word for it. But the cells up at the top that marked with a C, those are cells that are directly touching this competitor organism, whereas the NC cells are not touching. They're just only touching other Pseudomonas cells. And what you can see is that there's really not a huge difference. They're all able to turn on their type 6 secretion system. And in this particular image, it looks like maybe the ones not touching it are even turning it on a little higher. But let me show you what this looks in a graph form. So here we're again looking at type 6 levels on the y-axis, and we're looking at time, over time, how they turn on. So when the competitor doesn't have a type 6 secretion system, you don't see that much going on. When it has a type 6 system, now you see Pseudomonas is turning on. This is exactly what I showed you before. But the cells I'm showing you right here on this graph are the cells that were directly touching the competitor. Now let's graph the cells that aren't touching competitor. You can see they look pretty much the same. So it doesn't seem to matter if it's contacting this other organism or not. So this was a little puzzling. How does it detect this at a distance if it's not membrane, isn't getting punctured, it's not somehow feeling this other organism there directly? This was one of those moments where I had to really kind of step back from all of this and think hard about what is going on with Pseudomonas? What is it experiencing in these interactions where this competitor has a type 6 secretion system? Up to this point in my PhD, actually, I had really only been thinking about Pseudomonas attacking other cells. I hadn't really stopped to think about, well, what if the other cells are maybe also attacking Pseudomonas? It wasn't happening so much that it was really striking because I would have noticed all my cells dying if that was the case. But I decided to take a look and see if there was a difference in cell death of Pseudomonas when the competitor had or did not have its type 6 secretion system. So that's what this experiment is showing. And so you can see here that that's exactly what I saw. There's more Pseudomonas cells dying when the competitor has its type 6 secretion system compared with when it doesn't. Then we basically don't see any cells dying. So that's kind of interesting. This competitor is also killing Pseudomonas. So the next clue in solving this puzzle actually came in looking at how the timing of all of these events unfolded. So here we're looking at 3 parameters. The first one I'm showing you is the cell death of Pseudomonas, so the same thing I just showed you, but now you can see how it happens over time. And what you can see is that the Pseudomonas cells start dying almost immediately after you put them in contact with the Burkholderia cells. You see, even at 15 minutes, there's already been some cell death. The next parameter I looked at was the increase in type 6 secretion system levels. So that's just plotted on top of the other one. So you can see that type 6 levels start to go up, but only after some of the Pseudomonas cells have already started dying. There's a little bit of a delay there. Now, the last parameter we looked at was the death of the competitor organism, and that's shown here in these white squares. So here, this also kind of makes sense because you only start to see competitor dying after some of the Pseudomonas cells have turned on their type 6 secretion systems. And so this all kind of fit together. And it led me to wonder if maybe somehow Pseudomonas could tell when its siblings were getting killed. And somehow that was sending a message that maybe it's time to start mobilizing this defensive weapon and fight back. So before I show you the outcome of that experiment, I'm going to step back a little bit and tell you a little bit more about what we know about how the system gets turned on. So I told you we don't know what signal turns it on or when it gets turned on, but we do actually know already a little bit about the proteins that are involved in the turning on. So bacteria, of course, as I said before, don't have ears and eyes, but they do have these proteins on the outside of their cells that can detect changes in their environment, such as specific molecules. It's almost like smelling a little bit. And so that's this little cup that's pictured here. So a protein like this has to interact with a molecule outside of the cell, and then what it does is it sends a signal into the cell, and in this case, this particular one will then turn on the Type 6 secretion system. However, we didn't know what the signal was for this particular sensor. We didn't know what molecule it detected out in the environment. So I started wondering, what if there was something in a dead cell that was released that this sensor could then detect and send a signal into the surviving cells and tell them to turn on their Type 6 secretion systems? So this was something I could test directly. Just to remind you, when a cell dies, it spews out all of the stuff that's normally inside of the cell that can't get to the outside of the cell because of the cell membrane. But when it dies, all of that gets outside. It's kind of like its guts are getting spewed out. And we call this process lysis. So this is a cell lysing. So I could take a bunch of Pseudomonas cells and I could lyse them. I could grind them up and take all of their guts, and then I could add that to happy living Pseudomonas cells and see if this sensor protein could detect this. So that's what I did in this next experiment. So here we're looking at the activation of this sensor protein. And you can see that when you just have cells and you don't add anything to them, it doesn't get turned on. Now when I took these Pseudomonas guts, this lysate, and added it to the cells, you can see they get turned on really quickly. Immediately you start to see this sensor get activated. And importantly, when we took the guts of the competitor organism, the one that it's killing normally, you don't see any difference. So this makes sense. It doesn't really care if it's killing the competitor. That's the goal, right? But if it's getting killed, if some of its siblings are getting killed, that's when it knows, OK, now it's time to make Type 6 so we can fight back. So this was really exciting. I mean, we finally figured out a signal, an environmental cue that's turning on the system and is triggering the activation of the Type 6 secretion system. And we could basically think of these cell guts as a type of danger signal. When something terrible is happening in the rest of the colony, the Pseudomonas cells say, OK, this is dangerous. It's time to do something about it. So let's put this together and look at it how it would look in a population of cells. Well, I'm just showing you 4 cells here for simplicity. But the green cells represent the Pseudomonas. So a few of them get killed by this competitor organism. They spew out their guts. There's some danger signal in there that activates the rest of the population that's then able to defend itself by turning on its Type 6 secretion system. So there's one more exciting twist to the story that I want to share with you. So I told you that the sensor protein, we know it turns on the Type 6 secretion system. But the other thing that it does is actually turns on a bunch of other proteins in the cell. It turns on about 300 other proteins. And actually, a lot of them, we have no idea what they do. And we never really knew why this group of proteins was all getting turned on together by this particular sensor. But now we know that the sensor is responding to a danger signal. So we thought, well, it would make sense for the cell to want to turn on other things that are helping it protect itself. So what would happen if we inactivated the sensor and Pseudomonas no longer had the ability to sense danger? Would there be other ramifications of this? So that's what I did. So I'm going to show you 3 movies now. So in this case, we're looking at Pseudomonas cells dying. So these are the red cells, and they're going to be outlined in white as they die. So remember, that's just when they disappear from view. So the first movie on that side of the screen, you can see are just normal Pseudomonas cells growing with this competitor. In the middle, we have Pseudomonas cells where I've inactivated their Type 6 secretion system. So they've lost that ability to fight with the Type 6 system. And over here are cells where I've taken away their ability to sense danger. So they no longer have this, the sensor doesn't work anymore. So let's see what happens. So you can see the normal cells are just doing fine. They're growing. The cells in the middle, they're not doing quite so well. But over here, when they don't have the ability to sense danger, they are just getting decimated by this other organism. Pretty much every Pseudomonas cell that touches this competitor is getting wiped out. And remember, before, it was the one that was winning. So now it's just lost its ability to defend itself and to fight back in a much more dramatic way than just when it's lost Type 6. So what this tells us is that this pathway is more general. It's mobilizing a bunch of different things in the cell beyond the Type 6 secretion system that are helping Pseudomonas defend itself. So let's just think back to the spaceships for a second. So what I've basically told you is that if Pseudomonas, if one of its spaceships in its fleet gets destroyed by a competitor and it blows apart, the other spaceships can detect that debris flying through the air, and they know that it's time to not only get Type 6 going, but also to put up their defenses. And we don't know what those other things are yet. That's ongoing work. But potentially they're changing the outsides of the cells, their membranes, making them more resistant to things like toxins or antibiotics. And so this is a really exciting new area. There's a lot of new pathways that we can probably discover here. What does this look like on the cellular level? This is similar to what I showed you before. Pseudomonas, if it gets killed by a competitor, and I'll just point out, this killing doesn't have to be from Type 6 from a competitor. We showed that any kind of killing where the cells explode, where they lyse, will activate this pathway. They will then turn on the Type 6 system as well as a bunch of other factors that are also unknown, and then they're able to defend themselves. So this opens up a lot of new, fun, exciting research possibilities. There's all these new things that we now have an idea of maybe what they're doing, but we don't know how they work at all. So let's come back to what I started with yogurt. So this was my first glimpse into this invisible world of these alien creatures that are living all around us. So now, hopefully, I've convinced you that these bacteria, even though they're kind of primitive, we might call them, and they're single cellular, they actually exhibit some really sophisticated behaviors, almost like an immune system where they can detect danger, and then they know to fight back, which is something that cells in our body do. They also have this interesting ability to work together. It's not just each cell for itself. When they detect that something bad has happened in the colony, they're all ready to mobilize and get it together so they can survive as a whole. So this is sort of an interesting way that these single-cell organisms can almost behave a little bit like a multicellular group. But now finally, of course, what does this mean for disease? So I started by telling you that Pseudomonas causes really, really devastating infections for both patients with cystic fibrosis as well as people who have chronic wounds. And what can we learn from what I've just told you with these interactions with other bacteria? Why does that matter for humans? Well, actually, a lot of these infections are what you call polymicrobial, which means that there's a lot of different bacteria that make up these infections. It's not always just Pseudomonas by itself, and it's actually usually not. So maybe Pseudomonas is really good at fighting with other bacteria in our bodies, and that gives it a little bit of an edge. That's something that would be really exciting to explore. Another thing is that now we know that when Pseudomonas is exposed to this danger signal when some of the cells die, it goes into this other program of fighting back. So maybe our immune cells are killing a few of the Pseudomonas cells or some of the other bacteria in our bodies, and then that activates the cells to go into this danger mode. And maybe that protects them against antibiotics, for example. So most antibiotics actually are derived from other bacteria. We've just kind of stolen their molecules and used them to fight bacteria ourselves. So it would make sense for Pseudomonas to have the ability to fight back against some of these other antibiotics. So, I mean, a lot of these things are kind of years in the future. You know, it's going to take a lot of work to figure out if these things are actually happening. But I think that by thinking a little bit more about it from the perspective of the bacterium and from its interactions with other bacteria, we've learned a really important component of its behavior. And this can help us think more carefully about how we attack these organisms when they get into our bodies. So I just want to acknowledge a bunch of other people that were really involved in this project. I did not do all of this work by myself. First and foremost is my graduate advisor, Joseph Mougeot, whose lab I worked in when I did all of this work. And then I've listed some of the lab members that were directly involved in this project. And I also need to acknowledge a longtime collaborator, Paul Wiggins, who was very involved with helping us get the microscopy set up as well as analyzing a lot of that data. Thank you very much for listening.